Okay, hi everyone. I know we're running a little long, so I appreciate your sticking with us, and I'll try to keep this nice and on time. So yeah, my name is Mary Steele, and I'm a second year PhD student with Washington State University in the plant pathology department. I'm advised by Dr. Chakradhar Mattapalli, who I know you've heard some from already yesterday. And today I'm going to be presenting on one of the projects I'm working on, which is to determine the relative susceptibility of highbush blueberry phenological stages to botrytis infection. So we've heard a lot about botrytis today. I'm here to talk about a different aspect of it. And this is a project that was done in collaboration with Jeff DeLong and Virginia Stockwell. So I'd like to acknowledge first and foremost the funding sources for the project: Washington Blueberry Commission, the Northwest Center for Small Fruits Research, a USDA SCRI grant, and the WSU Department of Plant Pathology. And I'd also like to acknowledge that Fall Creek Nursery was kind enough to provide all the plants for this project, and we are very grateful to them. And of course, all the help from our lab members, in particular, Christina Ocaña Gallegos was very helpful with this project, and I couldn't have done it without her. So botrytis, we're all likely pretty familiar at this point. It's caused by fungi. It's caused by Botrytis species. Primarily, the species is Botrytis cinerea, and this pathogen is particularly tricky to manage because it produces a lot of airborne spores or conidia, which can then increase infection within a field. Botrytis as a fungal pathogen has a really wide host range, but it's particularly damaging for small fruit growers like strawberries, grapes, you name it, blueberries, red raspberries, blackberry, because it can cause, in blueberry, for example, blossom and shoot blight. So you lose out on that fruit if those flowers are blighted, right? And then also it can cause mold of fruit directly, both pre and post harvest. So as we've discussed some in both of these days, botrytis is conventionally managed with regular fungicide applications, and these usually happen for botrytis in particular when bloom begins to occur in the field, so usually between 5 to 10% bloom, and then those continue regularly through petal fall. So the question here is, when during the flowering stages are the flowers most susceptible to botrytis infection, right? So that was the objective of this project. We looked at stages between green bud, which we called F4, all the way up to stages where it's 75% blue fruit, so getting right up to harvest. And as I mentioned, F6, F7, which is full bloom, and F8 are the stages where management, so fungicide applications, are currently being targeted. So our hypothesis is, and it's been the hypothesis for a while with some previous studies in similar systems, that full bloom is most vulnerable to infection. And we wanted to test that hypothesis here. Another thing we wanted to look at is to see if there is any effect on removing senesced petals from green fruit, and if that could help reduce the chance of infection. So as we know, some cultivars have the tendency to hang on to those senesced petals. For example, Draper is a cultivar that tends to do this, and for that reason, we use that cultivar here. And our hypothesis is that these senesced petals, this dead tissue, is a type of tissue that botrytis might really thrive in, and it could act as a site of entry for botrytis, and therefore, if we removed those, that would reduce the risk for subsequent infection in the fruiting stages. To achieve both of these objectives, we are conducting screenhouse trials. So the first trial was this past season, 2024, and then we're going to repeat it in 2025. And these trials occurred at the WSU Mount Vernon NREC in a screenhouse, so enclosed but open air. And for the infection timing objective, we dedicated 55 Draper plants, again, thanks to Fall Creek for providing those. And we left 10 plants not inoculated, so those served as our control plants. And for the detaching petals objective, we dedicated 40 Draper plants. And on those plants, we had one cluster of flowers or fruit that were left uninoculated, and those served as our controls. And for the detaching petals aspect, we manually removed senesced petals at the green fruit stage from half of the plants to see if there was an effect there. And for all of our inoculations with the pathogen, we used one well-characterized Botrytis cinerea isolate, which we had previously obtained from a Washington blueberry field. So what we did is we inoculated clusters of flowers or fruit, depending on the stage, with a 10 to the power of 6 conidia or spores per mil. So this was a very concentrated spore suspension that we sprayed in the lab with a very small air blast sprayer onto individual clusters of flowers or fruit. To encourage infection if it were possible, we then incubated those plants in a 100% relative humidity growth chamber for 48 hours, and then those plants went back to the screenhouse for an additional 6 days. After incubation, we sampled from the screenhouse individual clusters of flowers or fruit, and we sampled them not only after that first incubation period, but we went back to the same plants every week until the end of the season, so until about 75% blue fruit. And with each sample, we surface disinfected the sample so that if there was any microbes or botrytis on the surface, that wouldn't count in our analysis. We only wanted to know what had reached inside the internal tissue. And we dissected the flowers into pedicel, so that's the stem leading up to the flower, receptacle and sepal tissue at the base of the flower, the corolla, so petals, and then stamens, and the stigma and style. And then we plated each of these parts, and if it was a fruit, we just cut it in half and plated the fruit and the pedicel onto quarter-strength potato dextrose agar, which encourages growth of fungus. And then any botrytis that we recovered, we saved and held onto as a stock culture. You might be thinking, there's a lot of botrytis out there, and since especially when we were working in a screenhouse, how did we know that the botrytis that we recovered was not just some other botrytis in the environment, right? And so to approach this, we verified the identity of any botrytis we recovered by matching it to a known fungicide resistance profile. So as we've heard, we've done a lot of characterization for fungicide resistance. And so we had this isolate that we used for this experiment. We know it's sensitive to fenhexamide at 10 ppm, but it's tolerant to fludioxonil at 0.1 and cyprotonil at 4 ppm. So all of the isolates that we collected from the project, if they match this phenotype, then we consider them the isolate we used for inoculation, and those were included in our analysis. So overall, this is just to give you an idea of the types of numbers that we were working with for this first year. I recovered 1,274 botrytis isolates from the screenhouse trial this year. And I just want to point out that some of those were control samples. So this number, 828, is telling you we had 828 flower and fruit parts that were not inoculated, so those served as our controls. And about 4% of those were positive for the botrytis isolate that we had used for inoculations. A smaller percent, about 3, were positive for some other environmental botrytis, and the vast majority, about 94%, were negative for any botrytis. We also had lots of flowers and fruit that we had inoculated, and so 3,558 flower and fruit parts were sampled, and of those, about a third were positive for the botrytis isolate we used for inoculations, about 2% were positive for some other botrytis, and again, the majority were negative for any botrytis. Okay, so this is a slide I want to spend a bit of time on. So this graph is showing you the relative likelihood of infection by botrytis at different stages. And this graph reflects entire flower or fruit parts. So as I mentioned, we dissected flowers and fruit and then plated them onto the medium. And these results are based on if at least one part was positive for the isolate of botrytis, then the entire flower or fruit was regarded as positive, and that's what's reflected here. The odds ratios show relative susceptibility relative to full bloom, which we set as our baseline because our hypothesis was that full bloom F7 was the most susceptible stage. The only significant difference we saw here after the first year was that the green bud stage very early on in the season was significantly less susceptible to infection than our baseline, which is full bloom. However, although these numbers look higher, none of these are statistically significantly more or less at risk for infection than our baseline full bloom. So what this tells us is right now we can't say that we saw any significant differences in susceptibility in any other stage other than F4 when we compared it to our hypothesis. So how about parts? Let's look at the parts of the flower. So as I mentioned, we dissected flower parts where we sampled, and here we see that petals were significantly more likely to become colonized internally with botrytis than other flower parts. So in particular, they were significantly more likely than our baseline, which here is the receptacle sepal tissue at the base of the flower, and more likely than the pedicel, which is the stem leading up to the flower. Another piece of evidence that petals might be a particularly vulnerable piece of tissue, especially when they senesce or turn brown and die, is that we did see a significant reduction in risk for infection when we manually removed senesced petals from green fruit. So this figure is showing you that when we inoculated plants at early green, late green fruit, 25% blue fruit, and 75% blue fruit, overall, we saw about 50% reduction in infection by botrytis, and this was statistically significant when we removed those senesced petals compared to when we did not. So our takeaways here, and again, I want to point out that this is just from our first year of screenhouse data. The data suggests that full bloom F7 is significantly more susceptible to infection than the really early stage of green bud. However, we were not able to parse out any other significant differences this year between F7 and other stages. We also saw that petals, live petals sampled from individual flowers, were one of the most likely parts of the flower to become colonized by botrytis, particularly significantly more likely than the sepal and the stigma and style tissue. And also we saw that when we removed senesced petals from the early green fruit stage, overall this resulted in a significant reduction in risk for infection, and this is something we definitely want to look into further. And with that, I know we're a little bit over time, but if there are questions, I'm happy to take them, or you can come find me after too.